In our previous lectures, we looked at the practice of science, and we looked specifically at how the practice of science leads us into biology and the main theoretical pillars of biology. Now what I want to do is start getting into really many of the details of the structure of biology. And like we said, it really starts at the cell molecular level in the way we're looking at it, and the concept of DNA is really being the foundation. So what I want to do is to start talking to you about cells and the major types of cells that exist. The earliest cells, some of the earliest cells that we know of, were of a type that we call prokaryotic, and I'll define that here in just a moment. But here's a picture of the early Earth. Not that early, actually. This is maybe about 2 billion years ago. And on the surface of the Earth at this time, 2 billion years ago, it was still quite a bit more volcanic than it is today. And there weren't insects, there weren't other animals, there weren't trees or plants of other sorts. There was this stuff that might have been sticking up over the surface. These things look like rocks, but they're not. They're called stromatolites. And what they are these big mats of these bacteria called cyanobacteria. Cyano refers to the color cyan because that's kind of their bluish greenish. And these things are photosynthetic. They're actually using energy from the sun to construct organic molecules out of inorganic molecules, and they are producing oxygen, and they're increasing the oxygen concentration in the atmosphere. I want to get into their structure. I want to get into the structure of these earliest types of organisms on the planet. Here's an example. This is not an early organism. This is a currently living organism. Right here is a bacteria. And if you look here, you can see, just follow the cursor there, you can see right along the outside this really dark line. What is that? Is that a membrane or is that a wall? The answer is yes, it is both. The membrane is actually something you can't very well see, but what you're looking at here is, in fact, the cell wall. So we'll discuss what the difference is between the membrane and the wall here in just a moment. But if you look inside of here, you'll see all sorts of other things. You'll see these dark dots. Many of these little teeny dark dots are these things in this cartoon picture, these sort of brown things. Those are ribosomes. We'll talk about what their function is here in just a moment. And all of this light-colored material is all DNA. And like we said in one of the earlier lectures, the DNA is not packaged in a nucleus in these organisms. So they are called prokaryotic. Karyo, that root, refers to the kernel of a corn. It's kind of a poetic way of saying nucleus. Prokaryo then literally means before the nucleus or before the corn kernel, literally. And that's pretty much it. There's really not much else inside these organisms. They're very, very simply constructed. They're extraordinarily complex physiologically, but their anatomy is not particularly difficult. So here are the key characteristics of bacteria, or prokaryotic cells in general. They don't have a nucleus or any other membrane-bound organelles. We'll see why they don't when we start talking about the other major type of cells, the eukaryotes. They have one big chromosome, this big circular structure here, and you can't really tell it's a circle here, but it is. And then these other smaller circular structures that are called plasmids. And so that makes up the entire content of DNA inside these cells. And the bacterial chromosomes, as I mentioned, tend to be circular. They don't tend to be linear structures. This contrasts greatly with the eukaryotic cells, which we'll talk about in just a moment. But I want to focus now on this. I want to focus on the cell wall and the cell membrane. What's the difference between the two? Now, the thing about the cell membranes, which we also call plasma membranes, is that every single cell has them. They're ubiquitous among all living creatures. You have them, trees have them, every organism on the planet has cell membranes. They're made up of a type of molecule, which is really a soap. It's called a phospholipid, and it also typically contains proteins that are embedded in that phospholipid soap layer. We're going to discuss the exact details of this later on in the course. But the thing you got to remember about this is this. They're made out of soap. So they are very thin. They are not quite as unstable as a soap bubble in the air because they're in water, but they act very much like actual soap bubbles, to the point where if you take two and put them together, they then will form one. That contrasts sharply with cell walls. Cell walls are very typically thick and rigid, and they're not in all organisms. They're in many prokaryotes, they're in many single-celled eukaryotes, and they are, by definition, in plants and fungi. But not in animals. By definition, an animal is not a creature that has cell walls. They're composed of various types of things. In plants and in fungi, they're made of carbohydrates, a type of sugar. In plants, they're a type of sugar called cellulose. In fungi, it's a kind of a rubbery type of sugar called chitin. That's pronounced chitin, not chitin. In other sorts of organisms, like for example the single-celled eukaryotes and the prokaryotes, they tend to be made of peptidoglycans, which is a fancy way of saying peptido meaning proteins and glycans meaning carbohydrates. But again, the key difference between these two plasma membranes and cell walls is that the plasma membranes act like soap bubbles. The cell walls tend to be rigid and give the cells their structure. Now, if you look inside of these bacteria, you can see these structures that I mentioned before. There's one, there's another over here, there's another over here. Each one of these is a ribosome. And this long, thin thing looks like a strand of hair is an actual DNA molecule, or probably in this case, sorry, an RNA molecule. The ribosomes are responsible for the most important process in all cells. What these things do, what ribosomes do, is they construct proteins. They make proteins. And they are the fundamental cell structures. In fact, it's likely that ribosomes arose before actual cells arose in the evolutionary history of life. We call them organelles, but that technically is incorrect. The technical definition of an organelle is something that has a membrane around it. This is not something that has a membrane around it. It's constructed of nucleic acid, RNA, and it's constructed also of proteins, about 60% by mass protein and about 40% nucleic acid. They are absolutely fundamental. So I do want you to remember that ribosomes are, in fact, things that construct proteins. Now, I'm about to show you a bunch of organelles, a bunch of structures within cells. What I want you to be able to do is two things. I want you to be able to recognize the structure in micrographs like this, or in cartoons, like, for example, this. I want you to be able to recognize them. And the second thing I want you to be able to do is to give a short description of their function. Like, for example, in this case, the ribosome's function is it constructs proteins in all known cells. There's going to be a bunch more coming, so be prepared. If we look at the bacterial DNA, like I said, this tends to be found in this large chromosome that's circular, and sometimes these little smaller plasmids. Here's an electron micrograph of DNA inside a cell, inside a prokaryotic cell. And this is not a double helix like we talked about before. This is not a double helix. What this is a bunch of coiled up double helices. You can't, in this type of microscope, actually see the double helix. But what you're looking at here is a coil of double helix, which is coiled on itself, which is coiled again on itself, and that's why it's thick enough to be able to see. That coiling and coiling and coiling is how prokaryotes actually package their DNA inside their bodies. And we call that process then supercoiling. So this is supercoiled DNA. It's a coil on a coil on a coil. This is not the way that eukaryotic cells package their DNA, and I'll describe that here shortly. But that's how bacteria do it. All of that contrasts extremely with this type of cell right here. What you're looking at here is a neuron of a eukaryotic organism. EU is a prefix that means true or correct. Karyo again means nucleus or poetically nucleus. So this is a true nucleated cell. And you can see in this particular picture that nucleus is highlighted in green. There's also all these other structures out here. For example, you see all these yellow strands and all these blue strands, for example, down here. The yellow and the blue strands are fibrils or fibers made up of actin and myosin, the same kinds of things that cause muscle cells to contract. They're in the cell as well. In fact, they're in all eukaryotic cells, and actually they define a eukaryotic cell. By definition, a eukaryotic cell is a cell that has a cytoskeleton. Now, you might have learned elsewhere that eukaryotic cells are cells that have a nucleus. That's okay, there's nothing wrong with that, except that professionals define eukaryotic cells as cells that have this cytoskeleton. The nucleus exists because the cell has a cytoskeleton. All the other membrane-bound organelles exist because they have a cytoskeleton. The cytoskeleton constructs those things, and that's why, as professionals, we tend to think of the cytoskeleton as being the key definitional difference between prokaryotic and eukaryotic cells. So the key characteristics of a eukaryotic cell is that they do have membrane-bound organelles, including the nucleus. They have a cytoskeleton, that's the defining property, and the DNA is packaged primarily in the nucleus. Not all of it is in the nucleus. Some of it's in the mitochondria and some other organelles, but most of the DNA is actually in the nucleus. And the chromosomes, as opposed to bacterial chromosomes, they tend to be linear structures, not circular structures. So if we look inside eukaryotic cells, one of the things that we see are these objects. Again, these are ribosomes. Ribosomes exist in all known cells because, again, they construct proteins, and proteins are the critical aspects of cells, the critical things inside cells that basically perform all the functions that the cell is capable of performing. They're the machines that do everything. They make up much of the structure of the cell as well. So the ribosomes are absolutely critical in making those things. In addition, however, they've got a bunch of other organelles that the cytoskeleton constructs. The nucleus, for example, there's ribosomes again. This thing called a rough endoplasmic reticulum, the Golgi apparatus, the smooth endoplasmic reticulum, lysosomes, peroxisomes, vacuoles, mitochondria, chloroplasts, and so forth. They also have the cytoskeleton, which is, again, the defining property, plasma membranes, and cell walls. They can. Animals don't, of course, but plants and fungi do. What I want you to do is this. You've got this here on the slide. You've got the list of all of these types of organelles and other structures, pardon me. For example, the cytoskeleton, the plasma membrane, cell wall are not organelles, but they're structures that I want you to know. This is also in your textbook. So this is what I want you to do. You've seen all these things before, at least most of them before. So this is really entirely review. What I want you then to do is to review these. Learn to identify each one of these structures in an electron micrograph like this, or in a cartoon like this, or like these. These are the cartoon versions. Your book has excellent examples of all of them. The internet also has excellent examples of all of them, but be very careful. Many of them are mislabeled. So again, your book should be your primary resource. But I want you to be able to recognize them in the cartoons, and I want you to be able to recognize them in electron micrographs. In addition then, I want you to know their functions. And you can see the functions are listed here. I want you to know these functions for all of these organelles, and I'm going to assume from this point on that you have these organelles basically memorized.